Competition is at the heart of all life forms. In reproduction, however, the competitors are less than 1/100th of a millimeter wide. In vitro fertilization, or IVF, made motherhood possible for women struggling to conceive by fertilizing a woman's egg in a dish and placing it back in the womb. But sometimes, regular IVF does not work well, typically because there are not enough motile sperm cells with healthy morphology. In this case, the doctors have to choose a single sperm cell to inject into the egg. But this procedure lacks the natural mechanisms in the woman's body, and it cannot be certain that the most adequate sperm is the one that fertilizes the egg. The problem is that sperm cells not only move fast, but are also nearly transparent, making them almost invisible under regular light microscopy. Staining the cells with visible color is not allowed in IVF, as it might damage the sperm, and the entire process is mostly manual and dependent on subjective decisions made by clinicians. Recently, Professor Nathan Shaked and his research team at Tel Aviv University have developed a novel optical imaging system that can visualize transparent sperm cells in real time, in 3D, and without staining. In contrast to regular microscopy, this system records how much light is delayed when passing through the sperm. This tiny light delay is impossible to measure in a quantitative manner using a regular light microscope. But if we record the addition of a low-power laser light beam passing through the sample and a clear reference beam that does not contain sample details, a digital hologram is acquired and is then processed to the optical thickness map of the sample. This quantitative map can be used to uniquely analyze sperm and other types of live cells in 3D without staining and without damaging the sample. The research team has shown recently that the new stain-free technique can be performed at the same level of accuracy as stain-based sperm analysis. Furthermore, the new technique is quantitative and allows obtaining a novel set of cellular parameters previously unavailable to clinicians, such as cellular mass and volume. Until now, such holographic imaging capabilities required expensive and complicated equipment and were out of reach for most clinics. But the research team managed to downsize the system to a small, simple, and inexpensive box that can be attached to a standard clinical microscope. Based on this novel approach, Professor Shaked and his research team are introducing advanced quantitative sperm imaging and automatic analytic capabilities in fertility clinics, providing doctors with more reliable information for IVF.